So I put up a short practice for us today. We haven't been really doing any of that A to T or A to U, C to G, G to C. I know we did it in 9th grade, but we haven't done it recently, or I haven't been having you do it this year. You did a little bit in your Wiser Me if you did that yesterday, but if you didn't do the Wiser Me yesterday, you didn't get a chance to do that. So let's open up in our Google World. I made a slide. It's only 2 slides. So you have essential dogma practice, meaning you will transcribe DNA into mRNA. You will translate the mRNA with the tRNA and the amino acid that it is carrying. So you should have a PowerPoint that looks like this. Here's your decoder box. So example, if I were doing this one, ACU, if this were DNA, I might need to shift this over. A bonds to T, right? They're complementary. T bonds to G and U bonds to A. My letters might be a little bit big. Let's go down to 30. Then A, if this was RNA, I don't have T available, right? So I'm going to use U instead. So A bonds to U, C bonds to G, and U continues to bond to A. So I forget who asked yesterday. So basically, is the DNA and the tRNA the same? Yep, except you substitute the U for the T. So if you have them being the same, you know that you're right on track. I'm going to try 28 for my font size. Ooh, no, 29. Okay, I'm going back to 30. Okay, now I got to find which amino acid this codes for. Am I going to use ACU to find my amino acid, or am I going to find UGA? Which one do I use to find my amino acid? Which is the actual code? Which one do I read? tRNA or mRNA?
Would it be mRNA?
It is. You read the message, right? That makes sense. The tRNA is only carrying it. It's not the code itself. Its code just makes sure it puts the amino acid in the proper spot. So you will see quite often FRQs that ask the job of each of those things. So it's really important that you know that the mRNA codes for an amino acid, the tRNA transfers it or carries the amino acid. So to read this, your first letter is here on the left. Sometimes you guys will see them in circles. So I'm going to go finding my first letter, which is A. This limits me to this row. I can only choose now from here. So it's somewhere in this column. Then the second letter you can find across the top, and this brings me down. So G, nope, C is my second letter. So now I want to limit myself to this box here. Okay, so where these boxes come together is right here, correct? So the third letter is U. So I read the third letter over here, the first row, the second row, the third row, the fourth row. So U is at the top. So of course they spell it all out for you, but if they didn't, that's how you would do it. You would go across, down, and then over from the right. So that narrows it down to the individual amino acid codon. And in this case, it doesn't matter which one you grabbed. This is called the Wobble Effect, by the way. So notice the first two are the same. The third codon is different, but still codes for the same amino acid. So they refer to that as the Wobble Effect. So the third position, so that tells me that this is coding for 309, THR. Okay. So in case you were not sure how to decode the box, that's how you do it. So you guys take a handful of minutes. You got 2 slides to decipher. This one, they give you the mRNA, work your way above and below. This one, they give you the DNA, so you just work straight down. All right, let's look these over. I spot checked a few. Let me know if there's any that you disagree with or you question. So if I'm spot checking here, UAU, U bonds to A bonds to T, except it will be U in tRNA. U bonds to A. So I'm going to look up UAU. UAU is tyrosine. So did we get 309, serine, tryptophan, glycine, tyrosine, phenylalanine, and stop? Excellent. Okay, next slide. So this one, methionine, your start codon, alanine, arginine, glutamine, I think, or is that GLU? No, glutamine, leucine, tryptophan, stop. Is that what you guys all got? The code right here, buddy. Remember to read the message. So in the last position, you're not reading AUC. You're reading UAG. UAG. So U across the top, carry it over to A down, and then G, the bottom position, is a stop. So make sure you read the message. It makes sense to read a message, not to read an, it doesn't even have a name other than anticodon. So good with that. You guys are ready to turn that one in. I did see one mistake on somebody's homework. The building and the reading. Okay, so remember we're reading 3 to 5 in order to build 5 to 3. Add free to the 3. So you're building 5 to 3, but that means you have to read 3 to 5 in order to be anti-parallel. So pay attention in questions. They will precede it with a 3' or a 5'. So make sure you're giving the answer in the direction that they're giving you, okay? Let's go back to our agenda slide. When we talked Wednesday, we were trying to decide what day would be best for the test. And I didn't think Tuesday would be good because actually I do have half of you in class that day. But I don't think it's fair to have some people in class and some people not in class. So we'll probably have tests on days when everybody is flexed or remote, which means we could do it on a Monday, we could do it on a Wednesday. So Wednesday is our first flex day of the new schedule. So we'll have our chapter 12 test that day. Today we're going to do information with mutation. I have an assignment that I put in that I think is kind of long. And I put it in under next week's schedule because it's going to be a carryover. So some of you haven't done the Wiser Me yet, so it would feel like a lot to do today if you had to do the Wiser Me and this. Those of you who already turned in the Wiser Me have our flex time available, so you have probably an hour's worth of time that you could work on this. But for those people who haven't hit Wiser Me, originally I wasn't going to give you anything extra, but just looking at this assignment that I really wanted us to do, because I think there's a lot of good stuff in it, I decided it would just be a carryover. So this is going to be today's work and Monday's work. And that way I can field questions Monday so I'm not going to lecture anything new, but I'll be available to help you with this concept. Does that make sense? So you want to be working on it a little bit over the weekend if you want. There's 2 parts to it, so maybe get the first part done. And then as you find you have questions, bring those questions Monday. Continue working. Maybe you're doing the second part on Monday during flex time, and I'm there to answer questions as you go. Okay. So nothing new is going to come up Monday. Monday's going to be a work day and a help day. If you have other questions about other things, we can do that too. So today we're supposed to cover mutations. So this is the same PowerPoint as we've been using. I did post a new PowerPoint in there for today just to give you some new content. That is actually a biology PowerPoint from a program that I purchased this year. And I just thought it was a real good, if you didn't have a handle of mutation, it was like an introduction. But so 2 big ones that the AP talks about a lot is cystic fibrosis and also sickle cell anemia. Sickle cell anemia comes up like 3 times. It comes up, we already did, well, actually maybe 4 times. It came up when we talked about protein structure, and it came up when we talked about genetics, and it comes up when we talk about mutations, and it comes up again when we talk about evolution. So be very familiar with the workings I'm going to hide my floating controls, the workings of this. While I'm lecturing, I have a tendency not to look at my chat box. So if you could chat Miss Mackenzie or throw it out to everyone and Miss Mackenzie will read that, that would be helpful. That would be beneficial to me. Okay, so now I can take up the box. All right, here we go. So talking about mutations, which you know are changes in the DNA, you did have some introduction to this already. So we're going to refer to these as point mutations when you make a single change in the genetic code, like in one position. That's a point. You made it at a single point. So they come in three forms, and you guys were introduced to this last Wednesday in your case study. Could be a silent, a missense, or a nonsense. So a silent mutation, and now that we're getting familiar with that decoder box, you can understand this a little bit better. Silent mutation still codes for the same amino acid. You saw 64 codons, only 20 amino acids, so many have the same codon that they code for. So in this case, here's our original code, right? We're going to make a point mutation in this position, and we might, like, you'll see to them referred to by number. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. This is in the 12th position. Usually your like formal resources, you'll be in like the 324th position. So in the 12th position, we had a C and we changed it to a U. So polymerase grabbed the wrong nucleotide and put in a U, and that's still coded for glycine. So there's no change in the amino acid. Therefore, there's no change in the protein's structure. Therefore, there's no change in the function of that protein, right? So when it talks about explaining, you want to go all the way back to the beginning, and you have all these cause effects that lead to the final result. Missense makes a mistake and grabbed the wrong amino acid, right? We made a mistake. So in this case, we changed, this is probably the 10th position. So originally we had a G and we coded for an A. And therefore, instead of glycine, we grabbed serine. Now, serine may have different properties. It may have different ionic interactions, sulfide interactions, hydrophobic interactions. When those different interactions take place, the protein shape changes. If its shape changes, it's no longer going to fit its substrate, and it won't be able to do its job. If that's an enzyme, for example. If it's some other protein and it's supposed to be a fibrous protein, it becomes a globular protein, obviously it can't work. Nonsense, well, that just doesn't make any sense. It's like stopping the sentence short, so the sentence didn't make sense. So the point that you change coded for a stop codon. So instead of having 324 amino acids in your chain, you have like 100. And so you don't have enough amino acid to make the proper protein to perform the proper function. So in this case, we made a point mutation in the 4th position. We should have had A, we put in a U. And when we did that, we ended up with a stop codon. So that stopped the translation too early, sorry. Okay, so try to figure these out, throw in the chat. Let's call them across the top. 1, 2, 3. So wild type usually is referring to the original. So you'll see it labeled different ways, or normal. So this is your original one, and this is your mutation. So 1, 2, 3, 4, what kind of mutation was made? This is your DNA, right? So CTT, and if we transcribe that into mRNA, you get GAA. That codes for glutamine. If we used our decoder box, our point mutation here coded for valine. So instead of glutamine, we got valine. So I'm hiding my controls. I'm guessing you said missense. So this is what happens with sickle cell. That's the exact mutation that happens for sickle cell. Here in the 6th position, notice you should have glutamine, and instead you get valine. So this is a recessive inheritance pattern. You'll remember that you have sickle cell disease and sickle cell trait. So sickle cell disease will affect a number of individuals, and it creates a lack of oxygen carrying capacity. Also, these sickled cells, they get stuck in vessels, so it creates a lot of pain. The sickle cell trait remembers that heterozygous version, and this is when it comes up with evolution, because the heterozygous version protects people against malaria. So it's more common in areas of malaria. Now, what causes the change in this shape is the fact that glutamine is a hydrophilic amino acid. So that means it's folded outward, right? It's going to be on the surface of the protein interacting with solutions. Valine is a hydrophobic interaction, a hydrophobic amino acid. So it folds inward, right? So this causes the hemoglobin inside of the cell itself to stick together, because now they're having different interactions, because different portions are on the outside, different portions are on the inside. So that's what's creating that sickling shape. So that's a single point mutation. A frame shift is when you take a letter out or you put a letter in. So that's going to shift the reading frame one position, or however many positions you added the nucleotides or subtracted the nucleotides. So in this one, we're shifting the reading frame, which means every amino acid after that will be coded for differently. Because now if we're reading every 3 spots, right? Every 3 spots are now off one position. So you might have inserted an extra base, or you might have deleted an extra base. So here you can see we're supposed to have U. This U has been deleted. So now it's going to read from this position on UUG, GCU. So it's 3 position, or it's a position off for each one. So instead of phenylalanine and glycine, we got leucine and alanine. Okay, so that adds, and it depends on that the one that is taken out could always create a missense. It could create a stop. Here's another stop where it's short. Those are the worst kind, the stop codons, or the stop, the missense. Sometimes, even though it's coding for a different amino acid, you still have the same interactions. So they have the same properties. So that won't cause much of an effect on the protein. If it's a nonsense, then it stops early. And so those are the worst type of mutations. So cystic fibrosis is pretty important, and it's going to be especially important now that we have more disease coming in, right? You might have seen the movie 6 Feet Apart, and so like we're all living the 6 feet now, but if you had cystic fibrosis, you couldn't come in within 6 feet of another cystic fibrosis person. So we did one time have 2 cystic fibrosis people in our school, and so their schedules had to be tailored so they weren't in the same class, nor would they pass each other in the hallways. So that was pretty important. And the reason why is because they can catch infection so much more easily, because they're not ridding the cells of their lungs of mucus, and mucus traps bacteria. So they get sick a lot easier. It happens 1 in 2,500 births, and 1 in 25 weight individuals are carriers. So what's happening is the normal allele codes for membrane protein. So this is our membrane and this is our protein, and it's a channel protein. So it should shuttle chlorine across the membrane. And if you remember, water follows salts. Chlorine is part of salts. So water would follow that chlorine across the membrane as well. So now you can't shuttle the water, and therefore there becomes a thickening of the mucus. Okay, so here's your normal functioning channel protein within the membrane, and you can see the chlorines moving out of the cell into the extracellular fluid. So water cannot leave either, and that creates a thick, sticky coat on the outside of the cells. So you have a mucus buildup in the pancreas, the lungs, the digestive tract, all increase your chance of bacterial infections. So people with cystic fibrosis have a shorter lifespan. They can live longer if they have treatment.
A patient with cystic fibrosis is susceptible to infections of the lungs. This is due to a mutation that affects a protein on the surface of lung cells. Many different mutations can cause cystic fibrosis, but all of them involve a transmembrane protein called CFTR that functions as a channel for chloride to move in and out of cells. In this case, CFTR is less able to transfer chloride ions out of the cell compared to the functioning protein in people without cystic fibrosis. The channel is, in effect, locked shut. This inability to transfer chloride ions has an effect on the outside of the cell. In people without cystic fibrosis, there is a balance of chloride ions inside and outside lung cells, which maintains the right amount of water and salt ions in the mucus lining the airways. Cilia normally sweep the watery mucus from the lung lining, keeping the surface clean. In patients with cystic fibrosis, chloride ions concentrate inside cells and draw in water by osmosis. With less water, the mucus becomes thicker and cilia can't move. Flow is reduced, providing an environment for bacterial infection. But there are now drugs that can help patients. To find a drug, researchers tested over 200,000 small molecules to see if any would bind to the CFTR protein. Then, for each small molecule that had some binding ability, researchers chemically synthesized different variations of that molecule and tested them for the ability to improve the function of some types of mutant CFTR channels. They identified a drug that, when taken by patients with a certain type of CFTR mutation, binds to the CFTR protein, opening the channel and allowing improved ion transfer. This increases chloride ion flow and maintains a watery, clear mucus, which helps the cilia move more freely and reduces the risk of bacterial infection.
Doesn't that almost like make you feel suffocating? I can't imagine. I mean, I can imagine because I've had like bronchitis when my lungs have been filled with fluid, and oh my gosh, I just got to think it's so uncomfortable. So I thought that video was pretty good, giving you a visual as to what's happening there. So we want to understand that a little bit. You'll see a lot of times on the AP test, they throw a disease at you that maybe you haven't read about, but you use these illustrative examples to find commonalities. So if you can understand how this works, then you can understand how something similar works. So you got the whole concept. The channels aren't working, chlorine can't move, therefore water can't move, and therefore you get a thick, sticky substance. So the mutation is in the protein that codes for this CFTR channel protein, or in the gene that codes for that. Here's an individual, she's getting treatment. They have to do like banging of the chest to loosen that mucus. There's a machine that kind of shakes them. They'll do that as well. And there's actually lots of different mutations that can cause that mutation in that CFTR gene. So it's not a single example, but there are common examples. So more than 1,000 have been discovered. The most common is a 3-base deletion. So you're deleting these 3 letters, which notice is going to create a frame shift. So now instead of reading ATC, this one's going to read ATT. And actually, that's just going to change one amino acid in the whole thing that way. It's not so much a frame shift when I spell it out. So just that one single amino acid is gone. And this is going to be in the worksheets that you do today and Monday. I forget what this is. This you can do on your own just for fun if you'd like. I just put it in there for interest's sake. Okay, so sickle cell anemia we've gone into detail about before. Today we hit a little more detail with cystic fibrosis. Over the weekend, you're going to learn much more detail about that mutation and that protein's function. If you haven't done the Wiser Me yet, you have that Wiser Me to do. Many of you, I opened it back up in case you wanted to make corrections. We're going to take our test Wednesday.